Okay, here's a podcast for a review of Unit 2A, which is standards 2.1 through 2.2, 2.3, 2.8, 4.2, and 4.4. So starting with standard 2.2, this is looking at cell parts and their organelles, and also looking at the role of the cell membrane in moving things into and out of the cell. So one of the things that you're going to need to be able to do is to identify some of the different cell organelles. So here we have a couple of different pictures of a plant and an animal cell. And you can see some of the major structures. So in the center of the cell you have your nucleus. Rough ER is the ER that has ribosomes attached to it. This structure here that looks like it's got squiggles on the inside, that is identified as a mitochondrion. The green ones here with these little circles inside, those are your chloroplasts. In a plant cell you have a very large central vacuole, whereas in animal cells over here it's much smaller. So there's several different parts that you need to be able to identify within a plant and animal cell. And so you have these pictures on the back of your review packet that you can take a look at. Now, not only do you need to be able to identify those different cell parts by sight, you also need to be able to tell the functions of them. So also in the review packet that I gave you, you have the cell parts chart. And the two columns that you need to concentrate on this particular one is the cell parts are listed over on the side here, and their functions are in this column here. And those are the ones that you want to kind of really concentrate on so that you can make sure that you know ribosomes make proteins. It's probably one of the most important ones that we've talked about. Cell wall is going to support and protect the cell. Nucleus controlling all the different cells' functions. Endoplasmic reticulum, depending on what kind you're talking about, you have smooth ER and rough ER. And they're going to be making and modifying and doing things to proteins and lipids. Golgi apparatus finishing off all your different proteins. Lysosomes breaking things down. Vacuoles for storing. Chloroplasts doing photosynthesis. Mitochondria doing cellular respiration. Your cilia and flagella are going to move the cell. And then don't forget, animals have a centriole. Plants have a centrosome, and those are used for cell division in a process called mitosis. So this part of your study guide you're going to want to take a look at, again concentrating on the name of the cell parts and their overall functions, what they do. Okay. You also are going to need to be able to describe the structure of the cell membrane. So remember, we talked about the cell membrane. We named a couple of different parts within it. So we always talked about how it has a bilayer made of phospholipids, and then stuck within that bilayer you've got proteins and you have cholesterol. And remember, some of your proteins are transport proteins, so molecules can go all the way through. Some of them are used for recognition, like this guy here has a carbohydrate chain attached to it, and that's going to allow the cells to recognize each other. Some of them are enzymes, like this one here might be an enzyme protein. You also have the cholesterol in there, which is strengthening the entire membrane. And remember, the phospholipid bilayer is that you have two layers of these individual structures, and each of these little things right here is an individual phospholipid, and they arrange themselves. And so the molecules, when you talk about how molecules can move through here, that's what our next part is. How does the structure make it selectively permeable? Well, remember that only molecules that are small and have no charge can diffuse directly across those actual phospholipids. Okay? Anything that is larger or has a charge to it has to use specific transport proteins to get across the membrane. And so those transport proteins are also specific. They're only going to let certain things in. And so this is how the membrane makes itself selectively permeable, because only really small things with no charge can go right across the phospholipids. Anything else has to use specific transport proteins to get in and out of the cell. All right. Next up is to differentiate between diffusion, osmosis, facilitated diffusion, and active transport. So you have this chart in your review packet as well. So again, passive transport, no energy, and we're going high to low, whereas at active, we are using energy, and we're going low to high. So you have your three different types of passive transport. And diffusion is, again, we're moving only our small molecules directly through the membrane. Osmosis is the diffusion of water. Usually that has to happen through proteins. And then facilitated diffusion, we're going to be moving larger or charged molecules through proteins. In your active transport side, we have some specific protein pumps that are going to move molecules against their concentration gradient to where we need them to. An example is the sodium-potassium pump. And then we have endocytosis and exocytosis. So endocytosis, remember endo means in, so we're going to take large amounts of material into the cell. And exo means out, so we're going to get rid of a whole bunch of stuff all at once when we do exocytosis. All right, standard 2.2 is comparing prokaryotes and eukaryotes. So you need to describe a prokaryote based on its cell structure. So remember, these guys are really small and simple. The only organelle that they have inside of them are ribosomes. They have no nucleus, okay, no nucleus inside. They do have DNA. They are always unicellular, and they always have a cell wall surrounding them. So these are super small, very simple, and examples of prokaryotic cells are all your different kinds of bacteria. Now we need to be able to do the same thing for a eukaryotic cell. Eukaryotic cells are much larger and more complex. Okay, most of them end up being multicellular, and they have a nucleus and they have lots and lots of different organelles all inside of them. So you can see they're filled up with a whole variety of different kinds of organelles that are going to do different jobs for this cell. So one of the things MCAS people like for you to be able to do is to compare and contrast the two of them. So here's another chart of that. Again, you can look at the things prokaryotes are missing or that are unique to them, and the same for eukaryotes. And the main thing that you need to note is that prokaryotes have no nucleus while eukaryotes do. And these are the things that they have in common. They both have cytoplasm, they both have cell membranes, ribosomes, because all cells need to be able to make proteins, and DNA, because DNA is the basic chemical of life. All right. Then we also did some classification of organisms into the 3 domains and the 6 kingdoms. Okay. So one of the things within here that we talked about was classifying things, plants and animals, between each other based on their cellular structures. So remember, there are 3 major things that plants have that animals don't. Plants have a chloroplast, cell wall, and that large central vacuole. And animals have centrioles. So those are your biggest differences. So if you look here, there's your cell wall, the centriole, the chloroplast, and then the vacuole is here, and you can just compare the size. It's much bigger in the plant cell. This one over here is the plant than it is in the animal cell. And over here is your animal. Okay, so they have a much bigger vacuole to store a lot more water. All right, you need to be able to name the 3 domains and 6 kingdoms. So the domains are archaea, bacteria, and eukarya, and they came up with the 3 domains based on their differences in their DNA sequences. All the bacteria used to be in one group, and then they started looking at their DNA and noticed that they were very, very different from each other, so they were categorized in a different way. And then your 6 kingdoms are archaea, bacteria, eubacteria, protista, fungus, plants, and animals. So you will need to be able to describe the 6 kingdoms. So you're going to need to look at this chart that is also in your review packet. And you notice that you have your domains up here. Oh, the domain bacteria is missing. This should say bacteria right here. There we go. And then the cell type. So these guys are prokaryotes. You have your eukaryotes here. One of the main things that they tend to have you look at is the differences in cell structures. So remember that eubacteria have peptidoglycan in them. Archaea bacteria don't. Protists, if they have cell walls, it's cellulose, just like plants. Okay, but they're always unicellular. These guys are multicellular. Fungus have cell walls made of chitin. And animals have no cell walls. So those are probably the things that you would look at the most, say like on an MCAS test or something. They would concentrate the most on the differences in the different kinds of cell structures. All right, 2.8 is comparing and contrasting a virus with a cell in terms of genetic material and reproduction. So number 1 is describing the structure of a virus. So a virus is a capsid that is made of protein. So here's your capsid going around the outside. And then inside of there is your DNA or RNA, depending on the type of virus that you have. And that's all that you have when you're looking at a virus. They are not made of cells. They can't reproduce on their own, and this is why they're not considered living things. Okay. Another thing that we looked at is how viruses use cells for reproduction. So all viruses use cells to reproduce, and they will eventually destroy the cell. If a virus does the lytic cycle, they will destroy the cell quickly, whereas if they use the lysogenic cycle, they actually become part of the host cell and then destroy it later on in the cell's life. Okay. Standard 4.2, we looked at both the circulatory system and we also looked at excretion, so the removing of waste from the body. So the circulatory system, we're going to describe how molecules get transported around the body. So remember, the purpose of the circulatory system is for transport of molecules. And it is your blood that is carrying nutrients, waste, and oxygen. And so when we look at the sequence of events that happen, so you start out, like here, they're starting out in the right ventricle, which is blood with very low oxygen. It's got to go out to the lungs, through the pulmonary artery, picks up oxygen, comes back to the heart on the left side, left atrium, down the left ventricle, out the aorta, where then it's going to go to all the different parts of the body, and the blood drops off its oxygen. Now you have low oxygen blood that's going to come back up to the heart through the vena cava, into the right atrium, and then we're back at the left ventricle. And so that is that sequence of events. And remember, blood always flows from the atria down to the ventricles and then out major blood vessels. Okay. We also looked at the excretory system in Standard 4.2. And remember, all the organs of the excretory system are going to receive their waste products from the circulatory system, because one of the things the circulatory system transfers or transports around. So the main job of your kidneys is to remove urea and water. Urea is a byproduct of nitrogenous waste from breaking down proteins. Your liver is going to help to remove ammonia and toxins that come into your body. Your skin can be involved in removing salt. Lymph can help you to remove bacteria, old cells, also can help you to keep your water levels in balance. And your lungs are involved with removing carbon dioxide. Okay. And finally, we have Standard 4.4, which is about the nervous system. Okay, so one of the things you need to do is identify a neuron and explain how it works. So a neuron is the functional unit of the nervous system. It is the cell of the nervous system. The dendrites are the small processes that come from the cell body, and they are going to receive the information. So the information moves in this direction, and then it moves down the cell body and all the way down the axon until it gets to the end, and then it goes on to the next cell. So the main parts, you have your dendrite, you have your axon, and your cell body. And remember, the impulse always goes from the dendrites through the cell body and down the axon to the next cell, and it always moves in that same general direction. Okay, we're going to look at how the nervous system regulates communication between the different parts of the body. So our central nervous system is controlling all of our different body functions in order to keep us in homeostasis, and it's made up our brain and spinal cord. So that's going to control all the different body systems, make sure our heart is beating and our diaphragm is contracting when we need to breathe and everything. That information that central nervous system actually has to be sent along the nerves of the peripheral nervous system. So your peripheral nervous system does two things. It receives information from the environment through the senses, and then it also sends that information from the central nervous system to all the different body parts, making them perform their certain actions, whatever needs to happen. So your central nervous system is like the boss, and it tells the peripheral nervous system what it needs to do. And the peripheral nervous system is also letting the boss know what's going on in the environment. So the kind of thing of the peripheral nervous system is communicating things back and forth, and the central nervous system is making all the different decisions. Okay, and finally we have describing a reflex arc. So a reflex arc is a very fast, predictable, involuntary response to a stimulus. So remember, it starts with your sensory neurons are going to pick up information about something happening, and it's going to send that information to your brain and spinal cord. The interneurons are what your brain and spinal cord are made of, and they connect the sensory and motor neurons. So they're going to send the message to the correct motor neuron. And then the motor neuron is then going to send that information away from the brain and spinal cord to the correct body part, telling it what actions it needs to take in order to keep the body in homeostasis. So here's just a quick picture of that showing you these different things. So here's somebody putting their hand on a tack, right? So there's your arrival of the stimulus. Okay, you've activated a pain receptor. That message is going to move down the sensory neuron into the nervous system. Here is your interneuron right here that is going to then relay that message over to this motor neuron, which is going to stimulate this muscle and tell your body or your hand to move away from the impulse.